Hi students, next important short notes is types of RNA from nucleic acid chemistry or molecular biology. This is also a repeatedly asking question. This is also already you read in your school days, but in first year MBBS biochemistry also it is very important. 5 mark short notes. You know all the three types of RNA, that is mRNA, that is messenger RNA, tRNA transfer RNA, and rRNA ribosomal RNA, right? Just 4 or 5 points for each with 1 or 2 diagrams is must for your 5 mark short notes, right? First one is mRNA, that is messenger RNA, which is always located in cytoplasm, right? Here, this mRNA act as it is synthesized as primary transcript. That is, what is the function of this mRNA? It transfers the genetic information from DNA to the protein synthesizing machinery on ribosomes. Right? Two words are very important. Genetic information from DNA to the ribosomes for the protein synthesis. For this mRNA, actually it is synthesized as an immature product only. It will go for some modifications, then only they will go active mRNA for the action, right? So here the modification, there are so many modifications. The two important modifications are poly A tailing and 5' capping. Poly A tailing means in the 3' end, that is the template strand of DNA, two ends are there. One is 3' end, another one is 5' end. In the 3' end, poly A tailing occurs. That means 200 to 300 adenylate residues are continuously attached in the 3' end, which prevents the attack by phosphatases and 3' exonucleases. Otherwise, if there is no poly A tailing, means the primary transcript or template strand of DNA will go for degradation or destruction of primary transcript. That is why poly A tailing modification is very important for mRNA. Second important modification is 5' capping. Here in the 5' end, there are 7-methyl guanylate which is attached in the 5' end, which act as a capping for this end, and this cap which prevents the attack from 5' exonucleases and phosphatases. So after these two modifications, the immature or inactive mRNA will be converted into mature or active mRNA, right? Second type of RNA is tRNA, that is transfer RNA. This is also the location is cytoplasm. Here the total number of nucleotide is 75 to 95, around 100. The shape of the tRNA is clover leaf-like structure. The leaf-like, it is clover leaf-like structure shape. It is leaf-like, and its function is translates the gene information. This tRNA, definitely you should draw one structure, right? You pick up the diagram from any of your prescribed textbook. That is why I don't want to draw the diagram for tRNA. You pick any tRNA diagram from any book, right? In the structure of this tRNA, we have 4 arms. One is acceptor arm, second is anticodon arm, third is D arm, fourth one is C arm, PSIC arm. Here what is the first one is acceptor arm is it carries the amino acid in an activated form to the ribosomes for the biosynthesis of proteins. Okay. This acceptor arm capped by a segment of CCA, that is amino acid triplet codon, in the direction of 5' to 3'. These amino acids are attached in this arm through phosphodiester linkages. These 2 points are very important for your description of acceptor arm. Second one is anticodon arm. We know the codon is present, triplet codon is present in the primary transcript only. While synthesis of protein, this tRNA go and combine with the primary transcript. So this is having triplet codon, that is called anticodon arm, right? With this anticodon arm, they will go and bind attached with the primary transcript of codon, triplet codon, right? Then only the synthesis will takes place. anticodon present which is complementary to the triplet codon on mRNA. This is the descriptive point. D-arm and C-arm, don't worry about the description. You just mention the name of these two arms only, right? But don't forget mention about the 4 arms names. Okay, bye. So students, we all know the types of RNA, that is rRNA, ribosomal RNA, mRNA, messenger RNA, and tRNA, transfer RNA. So RNA is a polymer of ribonucleoside which is linked by 3,5' phosphodiester linkages. Okay. Sometimes they are asking mRNA means you have to give full detail about the types of RNA, RNA and rRNA, ribosomal RNA like that. Okay. So what is ribosomal RNA? The RNA of ribosome is known as rRNA. What is the site? Actually this is something different. It is the site of rRNA is nucleolus, right? Some books say cytoplasm because next finally they will come to the cytoplasm for the assembly. That is why some books says nucleolus, some books says cytoplasm. But you mentioned both nucleolus and cytoplasm. Here the circular is shape is circular shape, whereas tRNA is clover leaf shape. This rRNA, it occupies 80% of the total RNA. That is the importance of this rRNA. Another important is, this is the structural and functional component of the ribosomes. This is about the introduction of ribosomal RNA. And what is the function of rRNA? This is the workplace for protein synthesis. That is where the assembly of the polypeptides takes place, and the process of translating mRNA, that is primary transcript, into the protein, that is protein biosynthesis, takes place in this ribosomal RNA. It is necessary to maintain the ribosomal structure. It also acts as some catalytic activities, that is by ribozymes and peptidyl transferase, right? So these are all the functions of rRNA. And what is the structure? That is, we have prokaryotic ribosomes and eukaryotic ribosomes. I am giving only eukaryotic. Here the size of the eukaryotic ribosome is 80S. Here the S means Sedberg unit. S means what is the unit of ribosome, eukaryotic ribosome. S abbreviation or S meaning is Sedberg unit. It is a measure of molecular size of ribosomes by ultracentrifugation depending on their sedimentation. Right? Ultracentrifugation is one of the separation techniques. So after this RNA is putting into the ultracentrifugation machine, depending on the sedimentation, they will be measuring the size of the eukaryotic or prokaryotic ribosomes. So 80S means size of the eukaryotic ribosomes. In this size, they have two subunits. One is large, another one is small, that is 60S and 40S. They contain more number of rRNAs and some proteins. This is regarding the structure of eukaryotic ribosomes, right? So you have to give some introduction about the rRNA and what are all the functions of rRNA, and finally you have to mention the structure of the eukaryotic ribosomes, and you have to understand what is S. S means one unit. Here the unit is Sedberg unit, which is a measure of molecular size by one of the important technique of ultracentrifugation. Okay.